Thank you. Can you all hear me? Okay. Well, thank you so much for the invitation, Brian, John, Francisco. This is a treat, really, to be here with you. So my marching orders were to talk about comparative phylogeography in the tropics. So, as a Brazilian, I could not resist to add the caipirinha in there. I hope you have fun along the way. And this is a multi-alter talk. I am here representing the Hacker Lab, Mega Lab at City College of CUNY. This talk is really made of efforts and contributions from several people from my lab and the lab of Mike Hickerson. Yvonne Prates and Alexander Zoe are here with us. They're two grad students in our labs. Diego Alvarado Serrano and Jason Brown are postdocs affiliated with our grants. And of course, Mike is here too. And I'd like to start by defining the scope of what I'm going to be referring as comparative phylogeography, as both Brian and Brett made sure to mention that we have lots of comparative phylogeographic definitions out there. What I'm going to talk today is really studies that have used information, aggregating information from multiple, genetic data from multiple species in the regional assemblage kind of level to infer how co-distributed taxa have responded to environmental shifts over time. And these environmental shifts could be abiotic, often the case for tropical studies, but they could also be biotic. And I love this picture, Don, and as captivating life because his work captivated so many of us and definitely captivated tropical biologists worldwide. But it took us a while to catch up. We're still catching up with the Northern Hemisphere, and that's exactly Brett's point very recently in his talk. Most comparative phylogeographic studies that we have in the tropics, they're recent, and lots of them, as Brett mentioned, are based on mitochondrial DNA information. This is just one sort of big show of how we're looking there. Basically, we have Craig Moritz in the wet tropics, and all of us trying to catch up. But I think collectively we've been doing a good job at generating data from these different and wonderful systems. There's a major common knowledge emerging out of these collective studies, and Brett already alluded to that, is that, and also Brian, that we have a lot of geographic structuring of lineages, irrespective of biome, irrespective of region of the world, really. And for tropical comparative phylogeography, people have focused a lot of common responses to climate changes, particularly glacial and interglacial cycles, which is this legacy of the Northern Hemisphere research that we sort of embraced and try to pull over as well. And the roles of rivers or valleys or topographic barriers. And usually these papers, when you read these papers, you can see that the authors are usually happy. They leave with the general impression that we find congruent responses, not congruent responses, we find congruent patterns in the distribution of mitochondrial lineages across species, or the patterns that we see make sense given what we know about the natural history. So these are sort of feel-good papers usually. This feeling changes as people started to collect nuclear loci. And these are all papers that struggle through Sanger sequencing of nuclear introns. And I think one of the reasons, Brett, that we don't see a lot of nuclear comparative phylogeographies out there is that it was very hard a few years ago. And we could barely generate these multiple markers for a single species, let alone multiple species, let alone at a community level that Mike Hickerson was dreaming of. But we're going to go there pretty soon. We are going there already. So there are significantly fewer studies, including information from nuclear loci. And then the few good, that good feeling about consistent or congruent patterns sort of goes away because people start looking at the processes that underlie those patterns, and they see that the processes don't match. Species seem to have diverged at different times. We have lots of pseudo-congruence, which was what Brett just alluded to. We have different drivers of lineage diversification, or just a lot of stochasticity in the data, which is not unexpected. Coalescence theory predicts stochasticity anyway. So we're slowly scaling up. These are the published papers that I found with subgenomic level data, or going into NGS. I know that lots of us are trying to generate that data. We're coming up. My apologies if your recently published paper is not there yet. The whole purpose of this slide is just saying that we're starting to move away from that Sanger sequencing and generating lots and lots of data from the SNP data sets. And we're all very excited about this. We hope to get increased resolution by this widespread genomic sampling, and freeing us from the nightmare of Sanger will actually let us go into more widespread taxon sampling as well. So this is a very exciting moment for all of us who can scale up at the community level. What these new studies are saying is that there's still a lot of idiosyncratic responses to former climate change. We have similar patterns. They don't seem to have been caused by the same process. So one of the goals of my work right now, it's involved with this NSF-funded Dimensions of Biodiversity program, and it's devoted to generate new data at the subgenomic level for communities in the Atlantic forest of Brazil, and combine and interpret that data in the context of what we know about the geology, what we know about the paleoclimatology of the system. An important issue in this project has been, how do we analyze this data properly, given what we're beginning to know about these former changes? So a lot of this effort has involved the generation of new methods that we're trying with these genomic data. And today I'm going to try to bring some of those results and share with you some of our thinkings and what are the promising ways that we're seeing this advance. So this is just an image to situate you. We're going to be talking a lot about the forest of South America, and this grant particularly has been focusing on the eastern coast of Brazil. This is the distribution of the Atlantic rainforest when the first European colonizers arrived in this area in the 1500s, and we barely have much left. We have about 10% of the original vegetation standing. So to give you a little background before we dive into this exciting new data, when we started this project, one thing that we decided to do was to pull out, let's gather all of the individual phylogeographic studies that have been done for this region, and let's see what we can do with these data. Can we find congruent patterns of phylogeographic distribution, patterns, phylogeographic endemism? Can we associate them with possible drivers or predictors of these geographic patterns? But one thing that became very evident to us is that we seem to have two different syndromes, two different types of biological responses in the forest. And they seem to have an association with where in the forest they were coming from. So the first thing that we did was an environmental analysis before even looking at the genes of how is the environment of this area that we called a single forest. This is a PCA analysis, a principal component analysis of the environmental descriptors of the forest. These are word claim points, pretty much, just describing bioclimatic variables of the forest. And what we see is that there's a very distinct, there are very distinct aspects between the Atlantic forest of the northern part of Brazil and the southern Atlantic forest component. These two areas are very different in terms of their average temperatures. They're very different in terms of the seasonalities. As a matter of fact, when we pulled all the phylogeographic data available for individual species for this area, and we just look at the species themselves and create distribution models and look at their distributions, we could see that a large number of species is basically distributed on this northern range of the forest. Another large group of species is only distributed in the southern range of the forest. And a few species, of course, are widespread. So we were interested that there's a turnover in climate, and that turnover is followed by a turnover in biology. Now, what we discovered is that not only these areas are different, but their predictions of how these different climatic spaces of the forest, if you will, responded to shifts in climate over time. They're also very different. So these are two maps that portray our, at that point, best hypothesis of refugia, forest refugia over time. for the northern spaces of the Atlantic forest and the southern spaces. So these are sort of the lowland, mid-altitude, warmer regions, and these are the subtropical, sort of montane regions. And as you can see, these actually were generated not with paleoclimatic data only for the Holocene and the last glacial maximum. These were generated with paleoclimatic simulations going back 120,000 years ago, and allowing for dispersal of the forest itself through time. But what we saw is that we find considerably larger predictions for stability of the forest over time in the north relative to the south. And in the south, it's like this montane forest refugia are scattered in, right now, mountaintops across the southern and the southeastern part of the country. What we found that was very interesting is that this distinct distribution of climates, both current climates and past climates, really help us understand how phylogeographic structure is distributed in the forest. In other words, you can predict or you can explain endemism of lineages within all these species differently according to whether they're in the north, more associated species associated with more lowland areas, or in the south, species associated with these mountain regions, in a way that if we had just looked at the entire forest at once and pulled all the data for all the species of the forest, we would see that both contemporary climate and past climates explain the phylogeographic endemism that we have in the data set. All of these 4 items, stability over the last 120,000 years, stability over the last 21,000 years, primary productivity and area as present-day descriptors of climate, they're all equally well predicting diverse endemism of lineages in the system. But if you analyze the north and the south separately, you can see that the drivers or the explanatory variables differ in the sense that the distribution of lineages and phylogeographic endemism in the northern range of Atlantic rainforest is very well predicted by how climates were distributed in the past. In the south, it's the other way around. It's present-day climates that limit the distribution of these lineages within species to certain areas of the country. So that was very exciting because, and it led me to think about temperature really rules. We really ought to talk about temperatures. It makes a lot of sense to think about glacial and interglacial cycles in the context of this forest. So we started this grant and we started to do what Brett was telling us. Go work with this climatologist, go work with this geologist. So the first thing that we did when we came up is that they showed us a map and said, look, all of this insistence that you tropical biologists have into looking at glacial and interglacial cycles and the influence of temperature in your case, they're kind of bogus. Here's what we think. And they showed us this map, which Chang published in a Nature paper recently. This map pretty much summarizes all of the proxy data that these people have based on cave deposits, pollen records, marine sediments, on how moisture has been distributed over time in these regions of South America. And what they are showing and what they're claiming is that all of the precipitation or the humidity patterns that we have in the northern Atlantic forest, they're different from those of the southern Atlantic forest. And I was very happy because I was seeing phylogeographic evidence of distinct responses. And they say, but this is not related to temperature necessarily, because we also have the same precipitation or humidity patterns that we see in the northern part of the Atlantic forest. We see them in eastern Amazonia. On the other hand, all of the precipitation patterns that we see in the southern range of the Atlantic forest are common with the Andean forests and the western part of Amazonia. So Chang et al. Proposed this new mechanism of moisture transport for South America that they really feel that explains a lot of the biogeographic units and the context across these major forest systems. What they are saying, and these are oxygen isotopes, what I want you to look at are the lines driven by the data, not the insolation lines. So the squiggly lines over here. So what they're saying is that whenever eastern Amazonia got wet over, time, this is a line that goes from now into 60,000 years ago, the same happened in northern Atlantic rainforest. At the same time, western Amazonia and the southern part of the Atlantic forest were dry. And the opposite happened in another time period. So these two areas that are here in pink are in sync, and they're out of sync with the areas in blue. And what they were saying is that this has happened repeatedly over time. So don't think of one glacial and interglacial cycle. Think of multiple pulses of forest contraction and forest expansion. They are differently in different areas of South America. So you can see here the expectation for instance, between 10 and 20,000 years ago, the expectation is that from the oxygen isotopes, that there was an expansion in the north and a contraction in the south. And these are data from fossil pollen data from the northern forest. Oh, this is much better. Showing that the northern forest indeed caught up with that. And the opposite is happening with the lowland, with the montane forest of the southern portion of the Atlantic forest. From 10,000 years forward, the pattern shifts, and the southern forest of the Atlantic forest expand where the northern forests go down. So Mahila Drew was saying, stop thinking about glacial cycles in the tropics. Think of these multiple pulses. They're saying, or they were arguing or suggesting that we think that these pulses really connected these forests, and they probably led to a shared diversity between the eastern part of Amazonia and northern Atlantic forest and the Andean fauna with the southern portion of the Atlantic forest, which makes a lot of sense for biogeographers. So one thing that we are doing in the lab, in collaboration with the lab of Mike Hickerson, is use and generate subgenomic level data from multiple assemblages and test this hypothesis of moisture dynamics. Do we see the patterns? Do we see the processes if we can use information about timing and spatial congruence to test these? So what I'm going to be talking about today is going to be slightly different from the two previous talks. It's going to have these new data that we're analyzing, and you can even help us think about it. These are fairly recent analysis. We're going to focus on space. We're going to look at three subgenomic data from 3 different lizards distributed along the Amazonian and the Atlantic rainforest. And we're going to focus on these 2 questions. Looking at space, how spatially congruent have been the range shifts in these species? And on time, how synchronous were any population syndromes of shifts in the population, and are they in agreement with this new idea of this moisture transport revolutionizing or changing the way we think about structures of phylogeographic endemism and structure in South America? So these are the data that we've generated for these 3 different species of lizards. Polychrus is definitely bigger. They're all 3 species associated with, each one of them occurs both in Amazonia and the Atlantic forest. Our sampling, you can see that increases this way. Polychrus doesn't have great sampling. We have much better sampling for Anolis ortoni and way better sampling for Anolis punctatus. This guy is a bit bigger than these two. These two within the same genus, they evidently have closer ecological characteristics. Anolis punctatus is definitely more abundant. It's bigger than ortoni in body size, so it seems to be doing better at exploring the environment. So we used genotype by sequencing, which is a RAD-like method, and generated a lot of SNP data for multiple individuals for these 3 species that we're now going to be analyzing. So the first question that we had is, how spatially congruent were these range shifts? And these are just very simple phylogenetic trees built with SVT quartets, actually, just to look at the structure of this data. And what we see is that there's a lot of structure in the SNP data. And for all of these species, we seem to recover the Amazonian clades as more basal, suggesting a source of the colonization from Amazonia to the Atlantic forest. This way. But we wanted to see, can we actually use information from these population genomic level information to infer this colonization rate? So one thing that we've been trying a lot is in our labs is to unite this thinking of spatial studies with coalescent and genomic level studies. So Diego Alvarado Serrano led a study to create this new method that is really looking at estimating something that he's calling the spatial site frequency spectrum, or spatial SFS. What the idea is here is really to capture information on what is the relative allele frequencies on the SNP data, and how are they distributed in space. So basically what you do is you have several sites, you use one individual per site, so very easy on your sampling. And you're gonna compare for every SNP, looking at the minor allele, where else in geographic space do I find that allele? So you're gonna compute that distance, the geographic distance, between your allele and where's that allele represented again in geographic space. So the idea is to do that for each site, for each locality, across all different loci. And what's going to give you there then is a spatial distance vector of the copies of that allele. So here's our vector for that site. What we can do is we can convert that vector that has those distances into a histogram. And this histogram is what we're calling the spatial SFS. Why is this histogram useful? Because it has been shown that the way that this histogram looks like, the irregularity of this histogram, can be associated with whether your locality is a source or whether it's been colonized later. So what this method is doing is really trying to look at the irregularity of the spatial SFS of this histogram to find, how likely was it a source or not. For that, Diego used Harpender's ruggedness index. So basically what it's doing, it's getting an individual per site, inferring the spatial SFS, and just looking at what is the amount of ruggedness that I have in that summary statistics. So we did that for the 3 species, and the numbers are so tiny that I put this ridiculously large arrows for you to see. What's happening here is that for both Anole species, the method actually recuperates the origin of this colonization event at the whole species level in Western Amazonia. So the method is picking up what those trees are sort of suggesting. Polycrus, it really doesn't. It suggests that the origin is in the Atlantic forest going into other regions. If you actually look at this ruggedness index, I don't know if you can read this, I don't think you can. This is just how the ruggedness index varies across the different locals that we're looking and localities that we're looking. And you can see that for these 2 species, this index actually drops very quickly, saying the index is doing a good job at identifying the source. For the 3rd species, you can see that it's not. So what we see here is that this very low level of spatial sampling is actually precluding our ability to say anything about this other lizard. What's interesting, though, is that you could run the same analysis just looking at the Atlantic forest. So if you look at another region, you're going to pick up different SNPs. You're going to pick up signatures of other colonization events. So what we did is we did the same analysis just for the Atlantic forest populations or localities. And what we find is that for both species, we actually have a very consistent site of origin of this colonization, which is actually at the border. For those of you, it's in the Espírito Santo, which is right in the middle of that big, large refuge that I had shown in the beginning. Here's the region for one species. That's where it is in that big context. So that's cool, right? We're having this information, combined information about space and combining information from your, spatial SFS, and recover information that, there has been some sort of colonization out of this refugia into other parts of the Atlantic forest. So what we learned from this part is that, do we have congruent spatial patterns for these species? Well, for the 2 species that we have sampled pretty reasonably well, yes, we have pretty congruent spatial signals. At the species level, both species left out of Amazonia, we can recover that with the spatial SFS. And within the Atlantic forest, both species had a very congruent site of origin of colonization within that biome. In the Atlantic forest, I was kind of psyched to see that the sources of colonization match hypothesis of the locality, or the placement, the spatial distribution of refugia that we had done in the past. And something that we really learned from this is that sampling still matters. Even with these gazillion SNPs, we need to go out and collect, which makes me happy. I know it makes Craig happy, because that's what we're here for. We're here to capture beasts in the field, right? So these results are inconclusive when you don't have enough spatial sampling. And this is very important to say for people who are still working in the tropics. Okay, what about timing? So spatial patterns are the same across species. Do we have the same when you look at timing of these colonizations or expansions or contractions? So remember that if this hypothesis is correct, our expectation is that over the past 10,000 years, what we had seen according to this system is an expansion of the southern Atlantic components and the western part of Amazonia. So an expansion of southern Atlantic forest and western Amazonian populations, if the populations were following. And a contraction in the northern Atlantic forest, and therefore in the eastern part of Amazonia. Do we see that when we look at our data? So what we did is we grouped our samples. We grouped our samples into sort of geographical populations, quote unquote. These are how they were sampled. We divided them into western Amazonian clade and an eastern, not a clade, a western Amazonian group and an eastern Amazonian group. And because of most of our samples were falling within what the climatologists had defined as the northern Atlantic forest, that was the only analysis we did. We did one analysis for Atlantic forest, and it's pretty much all the northern components. This was based on a structure analysis, because we want to make sure that we are not violating any assumptions of panmixia. So we made sure that we use our tree and a structure analysis that was done actually in SNMF to make sure that we are not violating assumptions there. So the first question that we ask is, do we have similar demographic syndromes across these different systems? In other words, do we have all the species expanding in one area and all the species contracting in the other area, just like they were expecting or now? So we ran, what we did is we simulated data sets with randomly drawn parameters under these 3 different syndromes. Going from the past into the present, we simulated populations or groups that were contracting in size, that were expanding in size, and populations that were constant. With those set of simulated data, what we did is we constructed site frequency spectra for each one of those simulated data sets. And what those are is really plotting the frequency of the derived allele and just saying what is the percentage of SNPs showing each different frequency. So we get this histogram. We use this as our summary stats. This is our currency when reevaluating our models. We then applied an ABC framework to this data. And basically what it does is, well, I've simulated data under these different histories, right? Now, let me compare all these simulated data to the data that I actually observe from each one of those groups. How do I do that? I put the simulated data through a funnel, in a sense. I'm going to say, how distant is this summary stat given a given simulation from the empirical data that I observe? I'm going to retain only those results of the simulations that are close enough to my observed data. So this is like my Euclidean distance here. This is defining pretty much how big or small is my bottleneck in that funnel. And I'm accepting a number of those simulation results. Those I'm going to use to estimate what are the parameters that are coming. Those are my hyper posteriors. They're going to help me understand what happened. What happened when I do that? Well, when we do that, the results are not at all what we expected. These are the results for punctatus, ortoni, and marmoratus. These arrowheads are indicating whether populations are expanding or they're contracting. What we're saying is that all different groups of punctatus are increasing, are expanding. All different groups for ortoni are going down, they're decreasing. And marmoratus just had enough signal for the Atlantic forest, is the same signal of ortoni. So we are living with a meh sort of conclusion. They said, no, this is not looking right. So can we look at the precise timing of these expansions or contractions. Maybe that can help us too. So what we did here is we used Xander's Wes and Mike Hickerson aggregate SFS, another SFS, to look at this information. What we're doing here is actually doing what Brett was mentioning earlier, aggregating information from SFSs for these different populations and analyzing them at a community level. This is the way that we can go forward into pulling data together from these very different communities. So what we're doing is we're collecting the SFS information from the different species. We're putting all them together in this multi-species SFS. But we are pulling out all of the singletons, all of the first bars for each one of them, and we're binning them. We're reorganizing them by size. That's our first column here. We're doing this for the doubletons. We're doing this for all of the alleles with different allele frequencies in here. This aggregate SFS then gives us an impression, gives us an idea of how this genetic variability is distributed across the entire community. What we did then is pretty much the same way that I had explained before. We simulated SFSs under different contractions or expansion syndromes, and we inputted in there different time periods for those events. And we converted them into these aggregate SFSs that we could then compare to our data. So these are the ways that Xander actually implemented this. We're simulating with a hierarchical model so that we have these hyperparameters that are global, the number of pulses of synchrony, how many times do you have those synchronic events, how many species are in that each pulse. You could have multiple different pulses within a community. The time of that synchronicity, but we're allowing parameters to be free for each species. So each species could have a different effective population size. Each species could have a different expansion or contraction magnitude. So this is, for instance, a simulation for expansion. This is a simulation for contraction. And once we have that, we do the same sort of framework in which you compare the simulated ASFSs with the empirical one. What do we find when we do this? Here's an analysis that is a multi-species analysis, including all regions for 2 species. The third species was such bad sampling for Amazonia that we couldn't do that. So this is sort of a 2-species global analysis. 2 species, all populations from different regions. What I'm plotting here is this dispersion index of the time of population size change, meaning this is the variance over the mean of the time. It's a measure of dispersion. How far do we have those measurements across species being from each other? If we had all species contracting or expanding at the same time, this dispersion would be very small, correct? We would expect everything to be sort of close to 0. Well, we have a pretty small value in there, but we also have a big fat tail going that direction. And when we actually put the posterior distribution of the mean time of the expansion, it's pretty large with a pretty big dispersal. So we're calling this ambiguous. It doesn't really look like it's synchronous. If we look only at the Atlantic forest, we can include marmoratas in that sample. So this is our global analysis for the Atlantic forest only. For those, it's pretty clear we don't have synchronous events. We don't have the populations contracting or expanding at the same time. That posterior distribution of that mean expansion time, it's not looking really good. Now, if we look within a species, this is orthonia only, within a species, I was actually happy to see this happening. Within a species, you have different regions, populations in the Atlantic forest are expanding at a very similar time that populations in the Amazonian east or west. This is actually the proportion of taxa in that synchronous pulse, and that's a pretty fat high bar around 1. So we were happy to see that, we have good support that when one species, everything is doing the same thing. And the mean expansion time, it's fairly recent. This is about 50,000 years for this whole thing. Assuming a generation time of 1 year, which is an iffy thing, so you can give or take. It's going to be in the late Quaternary for sure for this guy. So what are the main conclusions emerging out of this? Well, and how do we tie it with this recent, hypothesis of climate shifts in South America. We definitely see synchronous changes in northern Atlantic forest and eastern Amazonia. Everything that was happening in the Atlantic forest was also happening in the east of Amazonia. The problem is that, not the problem, but the reality is that whatever was happening in these two areas was also happening in western Amazonia. So this out-of-sync dynamics, as portrayed on this map, it's really not being confirmed or congruent with the data that we're seeing. What it means, we still have to wait and see. It could be that as we add more species, we're going to find that. It's just a sample. It's a very limited sampling at this point as we're testing these methodologies. But this could be also a neat example of this retroillumination process that you were talking about, Brett, that maybe we can tell the paleoclimatologist, really, how did you draw this line here? Is this some sort of eyeball interpolation? Because the things that we have there from here are definitely not. Maybe this area is more close to, the Andes. Maybe this area is narrower. So this is where we are now going back to all the paleoclimatologists. There are, Marie and Chang. They are working with us on this project and saying, this is not matching what we see. What could be done about that? It's also clear that we have these species-specific demographic histories here. And this is something that they are happy to see, because what they have been telling us is, you guys, in the neotropics, you probably had multiple breathings and contractions, expansions and contractions of this forest. So it's not that absurd to think that you're going to have different species reacting differently to these multiple pulses. It could be that idiosyncratic results are going to be the norm and not the expectation, differently from what you see in the northern hemisphere. And again, taxonomic sampling, both taxonomic and spatial, I think will help. And finally, there's no way out of thinking of species interactions. You have two species of anoles. Whenever one is going up, the other going down. The one that's going up is the one that we see more abundant today. It's the bigger anole in size. So it could be that there's something interesting to be looked at then. How am I doing with time? I'm good, right? Good. So that could leave some of us sort of sad, because, if we have different species reacting differently to climate change, how are we going to be able to put broad predictions of community-level responses to changes in the future? We don't think so. We actually think that we are an exciting time to think about that. And we've been trying to develop new methods to use information from this genomic or subgenomic level and genetic information to help us infer what species are going to do in their future climates. And this is work by Jason Brown, and he was in the lab last year. He just got a faculty position, so he left us. The idea here, and this paper is in press, is that we can use use molecular information to learn from the past, combining empirical genetic data and species distribution models, Brett, under paleoclimates, to estimate demographic parameters that we think are important as we predict responses to future climate change. Once we have those parameters estimated, we could go back to our simulations and estimate genetic composition of any geographical areas from the past to the present and into the future. So this is the long sort of phase one of this project. Imagine we can build species distribution models to estimate how suitable habitats were distributed in space. We have now ability to do this at least for the last 120,000 years. And we run forward in time demographic simulations with different priors or in the ancestral population size and different priors for migration rates and carrying capacity. We then run, I don't want to hit anybody in the eyes with a laser. We then run geographic simulation, coalescence genetic simulations back in time, matching our empirical data, and we do the ABC steps that we did to select what are the demographic parameters that are actually performing better given the empirical data that we have. Now, if we have these, we can go back to our paleoclimate models and do again forward in time demographic simulations with those demographic parameters that we picked, illuminated by genetic data, and then do our genetic coalescent simulations back, that would allow us to project the distribution of genetic simulation diversity now, and that would allow us to run forward and do them in the future. It sounds like phylofantasy, right? So we did this. We did this for a North American plant for which we actually had data. We did this for microsatellite data. This is the paper that's in press. 8 populations, 36 individuals. And we estimated with this method what is the amount of genetic allelic diversity that we have now and what is the projected amount of allelic diversity in the future. This is different from an SDM, right? Because it gives us not only a suitability of the habitat, but it actually gives us a guess of what's going to happen to the amount of genetic diversity that we have in that area. What we were happy with is that we picked this system because this is a system for which we had good estimates of dispersal. We had estimates of carrying capacity. So we could actually compare our ABC estimates, hyperparameters, to demographic parameters, to the empirical ecological data that are out there. And they were pretty good. So what we're doing now is we're doing this for the lizards, but we try to escalate this from going from macrosatellite to the SNP data. So these are the very first maps that we have of changes in allelic diversity, and we just got these, so we still need to work on it evidently. But by 2080, really, for Anolis punctata. So these are the species distribution models under current climate conditions and in the future. And from those, we estimated current distribution of allelic diversity, and then we projected. This is the difference. This is one subtracted from the other. These are all the conditions that we used. At this point, I don't put my hand on fire for this specific model. We're improving the way we're doing this. And the idea is that maybe we could do this for multiple species, and the even more ambitious trait is maybe we could do this at a community level. Maybe we could pull all data together and have estimates of genetic diversity for whole communities in the future. So final thoughts on this. We have an exciting way ahead. I think tropical comparative phylogeography will give a quantum leap in the next years because we're going to be able to pass the hurdle of genetic sampling and really dive into the subgenomic data across different continents. But what we need is really increased spatial sampling and increased taxon sampling. We are going to this community level phylogeography. We also need these analytical tools to handle all these data. So for all times, this is an exciting one to be. What we're seeing with the small analysis here is that just as we were seeing before with the mitochondrial data is that the spatial patterns are the same across species. And as we were seeing before with the limited nuclear data is that the processes are different. There is pseudocongruence. We have different processes underscoring these patterns. It may be a time to embrace these idiosyncratic results. It could be that there's more difference than similarity in some of these responses to climate changes in the past in the tropics. We still need to see this. The jury is still out there. But it seems like, we have increasing evidence of multiple climate pulses. And we now embrace stochasticity in our hearts. So it could be that this is going to change the way we look at comparative phylogeography in the tropics. And the last thing that I think we're going to is we've only addressed responses to the biotic environment, the abiotic environment. I think the next frontier is going to be looking at how species interactions are reflected in the genetic makeup of our populations. Of all places, the tropics are the ones that we really expect species to be interacting a lot. So with that, I'd like to thank you all for hanging in and thanking my lab and the Hickerson lab and all of the different people who have funded us through time. Thank you.